Well, good morning, everybody. I would like first to thank the BP organizer and Sue Burrell indeed for inviting me to present some new data on emerging therapies in the Porphyria. So you may remember that I was the head of the French Porphyria Center and also the head of the European Porphyria Network, EPNET. So let's go to the first slide. So we will start with the emerging therapies in acute intermittent porphyria. So you know, indeed, the actual therapy. All these actual therapy are developed to decrease LS1 activity in the liver. So you know, indeed, humanimine in the name or trademark of Normosan, said by ricordati rare disease, and also the effect of glucose. And there is a new drug now available, at least in many European countries. I don't know yet in UK, but I think it's going to be. This drug is called givosiran, and it's a synthetic small interfering RNA that reduces the expression of liver LS1 protein. So givosiran, I can talk about what happened in France. We have now around 25% under givosiran, 25 recurrent AIP patients, with frequent acute attack. And they are very well since 3 years. And I can tell you it works very, very well. So let's go to the emerging therapies. Emerging therapies are different because they are focused on the problem to increase the PBGD activity, which indeed is the background of acute intermittent porphyria with a defect in PBGD aminase activity. So the first one is gene therapy. You can increase the expression of deficient protein by using, you know that name now because of the vaccine, recombinant adeno-associated virus mediated transfer of a human PBGD DNA. So that's the gene therapy. Secondly, we have RNA therapy, which consists of administering human PBGD mRNA packaged into lipid nanoparticle. And then the third, we have the enzyme replacement therapy. So we can replace by using a recombinant human enzyme and administer it to and target it to the liver. And then the fourth is the chaperone therapy to try to recover some enzyme activity, but especially in special condition where with missense mutation, where the protein is not active but could be restored. So what is the first gene therapy for AIP? It was a phase 1 open label clinical trial using an adeno-associated virus, as I said, containing human PBGD cDNA. And it was done in Spain in 2015. And they recruited 8 recurrent AIP patients for that. Well, it was the first time in humans for the treatment of AIP, and I can tell you that the system was well functioning and administration was safe. But the problem is that it had a very moderate impact on clinical outcomes in patients. Less need for amine treatment. So you can see on this that this was the heme requirement of the 8 patients in the observational phase before the treatment. And after the treatment, you see there is a little decrease in the number of amine treatment needed. So, but it is not so very important. And the main problem, it did not succeed in reducing LA and PBG level in urine. As you can see that on these two figures, this is for LA, and you can see that it is the observational phase, and this is after the treatment, no change. The same thing for PBG. So it didn't work, but probably because the vector has to be improved for the moment. But we have no news about that. So, but I think people in Spain, they are still working on that, but we have no news for the moment. So what is the second? The second is the use of PBGD mRNA as a potential therapy for AIP. So we use something that you know, indeed. Again, it's like the COVID vaccine from Moderna. It's mRNA that it incorporated in lipid nanoparticle, LNPs, as mRNA vector, like that. So you have the mRNA inside, and around it's only lipids, constituted of different lipids. And this is very efficient to deliver therapeutic mRNA into liver cells. So here is the lipid nanoparticle. It goes into the cells here. It is endosomes, and then the mRNA is liberated, and then it is transcribed into the protein. So this is it seemed to be very efficient. Well, I should say that the studies are not done in human, but in AIP mice model. Just to recall you, we have a mice, which is called Vincent, because we saw Vincent van Gogh was probably porphyric. That's why this mice was called Vincent. This is an AIP mouse model. It's only a biochemical model. And there is no obvious acute clinical sign, at least. But you can induce the accumulation of LA and PBG and expression, indeed, by using phenobarbital, which is an inducer, as you know, probably. So if you induce the mice with phenobarbital injection, you can see an increase in LA and PBG expression in urine. So this is an old study, but it is very important that we have an animal model, because we need an animal model to test new treatments. So what does this lipid mRNA therapy in AIP mice model? It works very, very well. We have a rapid lowering of urinary LA and PBG. So you can see here. So we indeed inject the mice with the phenobarbital. So there is an increase in LA, an increase in PBG in red. And if you inject the mRNA of the human PBGD, then you have a very nice decrease in the expression of LA and PBG. So it works very well. And I can tell you that there are even more positive data on clinical manifestation in animal model and different animal model, again, and on rabbits also. So what about the future? Well, clinical studies should be done by Moderna in 2022, but it's still a question mark, because as you know, Moderna has a lot to do with the vaccine, and probably, but I think they will do it. At least we are still in contact with them, and they are still interesting. Indeed, there is a sort of competition between givosiran from Al-Nailam and the Moderna mRNA, but I think there is a place for both of them. So let's go we will see that in the future. So the third is enzyme replacement therapy, as we say. So here I will present something that was presented by a colleague in Spain called Antonio Fontanelas. And just to recall you the story of enzyme replacement therapy, because we had that in 2005, in 2005, sorry. And this enzyme replacement therapy was done by Zymenex, and it was called Porphyria. But the problem is that this didn't work at all. It was discontinued due to short half-life in the circulation and a lack of liver targeting. You can see that. You can see here the enzyme around liver cells. It doesn't enter the liver cells. So it doesn't work, in fact, and it is rapidly destroyed in the circulation. So at this time, it didn't work. So, but now Antonio Fontanelas in Spain has developed a new enzyme replacement therapy using conjugation to an apoprotein A1, which is a protein, and there are a number of receptors on liver cells, and you can conjugate that to the human recombinant PPG deaminase, and this enables indeed to an internalization of the enzyme in the liver cells, and you can see that this is the cells, and the enzyme is now in the cytoplasm. And so you have an enhanced hepatic PBGD activity. So this should be work. And as you can see, again, by measuring LA and in the phenobarbital treated mice, you can see the effect. So this is the untreated mice. She excretes a lot of LA, but when you inject the conjugate, you have a decrease of LA and you have a decrease of PBG. This is exactly the same as for the previous one. So I think this is a very interesting approach because it's not complicated. And there are also from Antonio Fontanella's, we have more positive data on clinical manifestation in animal model. And well, okay, the question is that will we have a clinical trial in human in Spain? I don't know. I have no answer on that. You know, it's always difficult to recruit patients and to obtain positive results from the health authorities. So we will see, but it's a very nice approach. So we can compare now all these analyses. So if we look at gene therapy, so the therapeutic action is delayed. It's only one single shot because you have immunogenicity. So you cannot do twice a gene therapy in one patient because you will have a vector immunogenicity. This is a big problem. So production cost is very high. And actually, this is not a success and we need new vectors. So what about replacement therapy during APOA1 PBGD? Well, this is therapeutic action is very rapid. Administration frequency could be weekly. We don't expect immunogenicity. It's very good because it's a recombinant human PBGD. So you should not have immunogenicity. This is applicable to sporadic and chronic presentation. It means acute attacks or recurrent patient. So production cost is really medium, not so high, in fact. Well, this has not yet been tested in human. So what about mRNA-based therapy? Well, this is a very rapid action. We utilize de-immunized mRNAs. Moderna is very good in doing that. There is a long experience, not only because of the vaccine, but because they have a long experience in developing mRNA therapy. So this is applicable to both sporadic and chronic, and the price is low. So Moderna is already developed in 2 diseases, OTC and MMA, the mRNA-based therapy. So I think we have good chance to have something very rapidly about that. I hope so, at least. So what about the chaperones? I don't know very much about that. This has been developed in Norway with Professor Sandberg, which is the head of HepNet now. So they published this paper very recently. Maybe you can do that. So the problem is that it's to restore partial enzymatic activity from mutated PBGD enzyme, but only those that are derived mostly from missense mutation. But as you know, missense mutation are the majority of mutation in patients. So it's a majority here. Indeed, it's not all mutation, but at least probably we can treat 40 patients like that, 40% of the patient like that. Well, I don't know if it works. For the moment, they have good results, but you know, the translatability to human is not yet done. So we have to wait until we have new data on that.